Welcome back, everyone, and welcome to the second session of the CCP-EM Spring Symposium 2021. So I guess in the interest of time, we should dive straight in and introduce our first speaker of the afternoon session. So it's my great pleasure to introduce Misha Kudrishev. Misha started out life as a physicist and did his PhD in Heidelberg, 4 postdoctoral positions in Basel, and he's been a group leader at the Max Planck Institute for Biophysics in Frankfurt. I believe he's going to Berlin later in the year, Misha. And he's going to tell us today about the structure of ion channels in membranes by cryo-EM. So take it away, Misha.
Excellent. Thank you for organizing this event. We really need more events. Great. So I'll be talking about some recent work, and I will get a little bit technical, but this is all just to share how we try to look at structures of membrane proteins in membranes, mostly by cryo-EM, with a focus on ion channels. So in general, the research of my lab is about bringing membranes into membrane protein research, because membranes often affect structures and functions of membrane proteins in non-trivial ways. It's not only that the lipids affect, depending on which lipid is present, the protein may be in a different conformation, but also transmembrane gradients affect conformations of proteins, and also sometimes proteins could organize lipids around themselves in non-trivial ways, potentially promoting cooperativity. So it's possible to do very pretty structures at high resolution, and they're very relevant in nanodiscs. But so today I want to focus on tomography project that we have been looking at a structure of ryanodine receptor in native membranes. And I'll guide you through our workflow, how we try to get to higher resolution from subtomogram averaging. So the biological process is excitation contraction while during the muscle contraction. So when a signal comes from a motor neuron to the muscle cell, it generates an action potential over here, which then goes into an invagination of the muscle cell, which is called the T-tubule, where it activates voltage-gated calcium channels, also known as GHP receptors, and then calcium flows into the cell. This calcium activates calcium regulated calcium channel ryanodine receptor together with a physical interaction with the voltage-gated calcium channel. And together, this causes even more calcium release from the SR, which then leads to the actin myosin fiber contraction. So the structures of the key players have been solved in their purified forms, but we actually wanted to look at the structures of the supercomplex and how they interact inside the native membranes. And obviously, it would be super exciting to visualize how the physical interaction of the 2 channels leads to cooperativity and to the function. For this, we decided to go for taken a rather old protocol, actually, and we take fresh rabbit muscle, and with a rather simple biochemical procedure, we homogenize and make rounds of centrifugation and 2 sucrose gradients. After this, one of the fractions goes directly to the EM grid, and we can image it in a cryo-electron microscope. So this is a tomogram, so it's a tilt series where we tilt around the tilt axis, and we see essentially vesicles and gold beads. After this, we generate tomographic reconstruction, which is a 3D volume, and the slice through this tomographic reconstruction, we can see individual molecules if they're large enough. So in this case, ryanodine receptors are 2.2 megadalton proteins, and so sometimes we can see them next to the membrane that could be the T-tubule. We can then obviously pick multiple receptors and mutually align and average them. It's called subtomogram averaging. And we could get a structure at 12.6 angstrom resolution from roughly 2,700 particles applying the C4 symmetry. Unfortunately, so far, we could not get enough particles to be able to see the membrane of the presumable T-tubule attached to it and the potential interaction with the voltage-gated calcium channel. But the structure that we did get actually allows us to position a rigid-bodied dock the available single particle cryo-EM atomic models from single particle cryo-EM obtained by, let's say, Joachim Frank's group. And so what we could see is several additional densities, but what's most interesting for us was that the membrane is actually not flat, but it's a little bit bent, and the curvature of the membrane is different to what would you expect from a curvature of the vesicle itself. It's much more curved. And you can actually also see the curvature of the membrane around individual ryanodine receptors in tomograms. So it's possible to activate the receptor by adding calcium and plant toxin ryanodine, which will lock the receptor in the open conformation. And we've done the structure. So here you see the toggle between the open and between the structure in the open state and the closed state. And interestingly, the structure, when it's locked in the open state, also has an even higher curvature of the membrane. So this means that while the receptor opens, it actually also bends the membrane, and then the membrane wants to bend back to the equilibrium state, and which obviously applies extra additional force to the open receptor in the direction of closing it. Here we had less particles for this receptor, and we obviously got a little bit lower resolution for this. So a couple of extra densities, we could see that inside the sarcoplasmic reticulum, there are extra extensions which follow C4 symmetry. And there is also something that looks like a transmembrane helix, which does not seem to belong to the receptors individually. So we attribute those densities to the roughly 200 residues that are not tracked in the atomic model, which probably are only present in the not so well, in a not so stable form when the protein is purified. But so what's interesting is that we also did classification into 4 classes. When the purified receptors were classified into 4 classes, the occupancy of those classes from 2 groups were very similar, so roughly 25% in each class. But we actually had one major class where roughly 45% of particles went, and other classes were minor. So which suggests that maybe there is a different conformational landscape of the receptor when it's present in the native membrane. So in native membranes, it can contain interesting combinations of lipids. And so obviously, for some reason, it also bends the membrane, and we don't understand why so far. But so in order to understand this a little bit more, we would want to have all those classes actually to be at higher resolution. And that a higher resolution would, let's say, if we get to resolutions of 5 to 9 angstrom, we would have an opportunity to resolve secondary structure. The protein is mostly alpha helical, and we could be able to at least flexibly fit the available atomic models in order to understand what are the conformations of those of the protein in different classes. So we decided to invest a lot of effort into going from those structures, which this one is at roughly 13 angstrom, and those have lower resolutions because they have less particles. And so we thought about tomography, and we came up with this idea of hybrids, subtomogram averaging, and single particle analysis. So this comes from a notion that when we record a tomogram, our untilted images are recorded first, and then they are actually they have lower efficient thickness. So essentially, the best data we record in the untilted image. So we just decided to use high exposure during the untilted recording of the untilted image, then incorporate this image into the tomogram. And as a result, we would be able to perform the reconstruction only from the untilted image eventually, with the potential to refine the particle that is in the image against the 3D structures. So for this we use RelyOn, and this has been actually very, very effective for the large ryanodine receptor. We would get 13 angstrom with a regular subtomogram averaging, which I think is a very nice structure as well. But with the further refinement, we could get it to 9.1 angstrom resolution, and that structure actually showed rather well defined those sausages, which obviously are alpha helices. So essentially what we do here is we do a 3D reconstruction from a single particle reconstruction. Unfortunately, we still do need tomograms, because if we don't have tomograms and only try to pick up particles in 2D projections of native vesicles, we didn't get it to work very well at all. And the limit of the resolution here is roughly the number of particles which are limited per tomogram, because it's not a very abundant protein. So we have roughly 2,500 particles with symmetry, and I assume that the limit of our resolution is actually directly number of particles. So but we're very happy that we can see most of the alpha helices, and we believe that we could refine flexibly the available atomic models into these maps. So all we need to do is now have several structures that would characterize the conformational landscape of the receptor in this setting. And so that's what we decided to do. Venbo, who actually collected, Venbo Chen, who recorded this dataset, left at some point by graduating from his PhD, and we turned, it actually, even very good students actually experience very steep learning curve, as subtomogram averaging does have multiple manual steps, and it takes significant computational time to store data and to process the data. And as a result, I will use this EMDB statistics that even though the structures do get better over time, they're really, really difficult to produce. So obviously we need to make tomography a little bit more accessible and potentially slightly higher resolution, also for labs who actually do tomography full-time like ours. And so the consideration of throughput is that tomographic data collection is slow. If you want to record a nice tomogram, it takes us 15 to 20 minutes. And there are fast schemes, which I'm very excited to start using hopefully soon. And we will have a talk about this in an hour. However, the problem still is that once we do record our 1,200 tomograms in a weekend, we still will need to process them. And in our experience, processing is partially manual and is rather cumbersome. There are reported workflows, but we never got them to work essentially automated. So what we try to do, we try to break down the workflow in the blocks and see what is the best solution for a given block. And it turns out that actually most operations do not require human intervention or could be done automatically or semi-automatically, so to say. Right, and so Nikita is a computer scientist, and so he put a lot of effort into putting the one operation after another in a workflow that a person can use. And so most of the steps we can now automate. I will show you an example later. So the weak point here is the tilt series alignment. There, if the tomogram is the tilt series are not very well aligned, then the tomograms will be misaligned, and then everything else you do with these tomograms will also be poor in resolution. And then the particle picking in 3D tomograms is actually more difficult than one could think. So that's what Nikita did. He programmed it's a config file in the JSON format, and then he makes one block of steps after another. It has default parameters, and it mostly works. So as a highlight, there is a new functionality in Dynamo for tilt series alignment using gold bits, and it's scriptable, so it works very well. Excellent tool. It does fail from time to time, but then if it does fail, we know that it fails, and then we run another solution instead from iMod. So it's essentially a combination of several different tools, and that are all brought to the same input-output formats and potential to actually record what the person have been doing with the data. It's written in MATLAB, but it also runs without MATLAB. So essentially, you don't need a MATLAB license if you want to run it on, let's say, on a cluster. Okay, so these are the keywords that we came up with, and so it's, we decided that it has to be a tomo bear. So we call it this way from now on. Okay, and then we decided to benchmark the tomo bear, and that what the whole community does is essentially processing the GAG data set from John Briggs's group. It's like the apoferritin of tomography. It's 43 tomograms they record on the K2 in super resolution mode, but usually people process the counted mode of it. And it was processed originally to 3.9 angstrom, and it went all the way to 3.0 angstrom recently with warp and AM. So Nikita essentially programmed a workflow, and then it ran on a single machine in 31 hours. So the right column is the accumulated time. And so what it ended up with is a 7 angstrom structure that was obtained almost automatically. And so most of the time was spent for template matching of the template against the tomograms and of the subtomogram averaging process that we do in Dynamo on beamed data. So after someone reaches 7 angstrom, we think that at this point, subtomogram like automated routines stop working, and the user has to take over and actually to further refine the structure. So the way we judge, so one of the ways we basically look at the structure, the resulting structure, but one of the ways we judge it is by the way how the particles that are in the final reconstruction fit into the geometry. So here, this is an example where it fits rather well. Maybe some of them are still a little bit, maybe some of them are still rubbish, but the user has to now take it and clean the dataset further in order to get higher resolution. Roughly 280,000 particles came from 43 tomograms. So that's why the processing time, and even on a powerful box, is rather significant. Okay, so at the high resolution end of our workflow, we basically would have to get unbeamed 3D particles from unbeamed tomograms. They are very, very large, and especially on the K3 with a very large chip, they become prohibitively huge. So what Ricardo Sanchez in our group did, he introduced substack analysis, which we call Susan now. Essentially, what he does is he replaces the order of operation when we calculate cross correlation between 3D volumes. So usually you would rotate your reference and cross correlate it to particles with taking mismatch into account. And for this, you do Fourier transform. So essentially, you do a lot of 3D rotations and Fourier transforms of large volumes. And this is very expensive, even on the modern day graphical processors. So instead, Ricardo replaces the rotation of the reference with the projection of the reference in a given in a given set of angles, and then he calculates 2D cross correlation between the projections of the reference and the particle which is observed in different images of the data. So this is essentially allows to replace in 3D Fourier transform with a series of 2D cross correlations, and it has lower computational complexity. It has a couple of perks, such as you can apply CTF, the exact defocus and B factors for dose weighting directly for every projection in a substack. And then you can also play with excluding certain projections or in a very, very streamlined way. It's a very efficient implementation. So essentially, he never needs to extract the particles anymore, because he can load the tomogram in the memory of the graphical processor and on the fly extract all the particles from it. And if you have, let's say, 10,000 particles in a tomogram, which I'd love to, then you essentially do not need to generate any additional files. This is very quick. Yeah, and so the runtime for Dynamo, if we increase the box size, which you have to do if you want to get a very high resolution, you have to have a very small box, and then obviously the size of the box increases. Susan, this is in logarithmic scale. Susan performs up to 1 or 2 orders of magnitude faster, and then you can do calculations that would be prohibitively long to perform on the classical, using the classical algorithms. So we released this, the basic idea of projected-based cross-correlation in 2019. And so Ricardo has been working on making a tool, which we now have. So this is the GAG data set. We could essentially, by not doing the unbinned reconstruction, but having the original, just the stacks, we could go slightly below the counted Nyquist. So this is still a preliminary map. We're still working on this because before we release it, we need to make sure that we can control overfitting, which we potentially may have. So yeah, but it definitely is a very good map. Okay, so now equipped with all these cool tools and the bear, Venbo and Vasily, who now picked up the project, recorded a large data set, roughly 300 tomograms, hybrid dosimetric over 5 days. And then, so I would say it's a huge data set, but with the help of the automatic TomoBear enhanced processing, we actually could process it in reasonable time. And so the only thing that the user needs to do essentially is to make sure that the gold beads refinement is well done. Usually it is. If it's not, then a couple of gold beads, maybe a couple of contours need to be added or deleted. And then what we do, we oversample huge amount of particles from cross-correlation volumes. I'll show you how it works. So this is a template that we use, and this is a tomogram. It's without any filtration. So this is a very nice thin tomograms, and this is a cross-correlation volume. So we see peaks also from other things like gold beads, edges, membranes, and this is a cross-correlation for a good tomogram. So from here, we would obviously pick mostly the receptors. However, if the tomograms are thicker or have less projections, then it's not as pretty. So that's why we do extract 1,000 peaks from a tomogram, and then we have 300 tomograms. So Vasily broke down the data sets in several batches. And so for each batch, he does a multi-reference alignment and classification. So this is our initial template. And remember, we super oversample it. So at each step, he just throws away the particles that are not classes that obviously are not very good. And he does it very, very thoroughly. So like here, he takes this class and then classifies the particles from this class into another classes. And so actually here we see that this looks like there are good particles in there. And then he takes the rubbish classes and still tries to recover the good particles from those rubbish classes, after which he ends up with roughly 3% of the initial selection of particles, which actually are mostly particles, and we can look at them and see that they are particles, right? So we only keep those, and then we refine. We first do subtomogram averaging with lower binning, and we get roughly to 14 angstrom from roughly 4,500 particles. And then when we only use the high dose image from high dose refinement, we use Reliant to the local refinement, a very nice reconstruction. We lose some particles on the way. We end up with 10.0 angstrom resolution. It seemed to have the hints of secondary structure, but actually the map here is on the left. It looks a little bit worse, and actually it's much more difficult to see secondary structure alpha helices in this map as compared to the previous map, which is displayed on the right. So we need to add a bit more data and play with the classification. This is work in progress, but yeah, this is essentially how roughly in half a year we could get this map. So this was my last slide. I hope I could show you that we can do structures of ion channels in native membranes, and they seem to be slightly different at times from the structures of purified receptors. This includes extra densities, bent membrane, and we could get structures at sub-nanometer resolution. And with these structures, we could see some secondary structure. Then I'm really proud that we built this little system. We'll want to try to release it. It improves processing time, and also it also introduces an in-the-lab standard where I as a PI can go and see what the person is actually doing, and colleagues can also exchange experience. It's easier to troubleshoot. Tomography occupies a lot of space, and here we have a functionality to actually clean up the unused intermediate steps. It has the potential to run live in principle. We need to redo the data structure a little bit, and then it can also, and it could, so I hope it will, prepare depositions, write method sections for the paper. And so little greed that I have to admit that we probably would have gotten more particles if we didn't go for very high magnification, because very higher magnification results in lower field of view. So yeah, I think this is on the experimental design side. So sometimes me and other people maybe should consider not being so greedy for the mag, especially when it gets to tomography. So we took this picture today. So it's a sunny day in Frankfurt. I thank my funding and the people who actually did the work. Ricardo developed Susan. He's here on the left. Vasily over here did work on Randin receptor with Van Gogh, who already graduated and left the lab. And Nikita, he's not in the picture, developed TomoBear. I'll be moving to Berlin, yes, in August, and there will be a lot of bears which try to make the letter T on campus. So if you want to do something in tomography of membrane proteins, write me an email, and thank you very much.
Thanks, Misha. A lovely talk. There are lots of questions, so I'm going to cherry pick some and then leave some of them for you to answer afterwards. So first off is an easy one from Louisa. Did you do anything to get rid of the excess sucrose from your purification? Because you're at 30% sucrose. Did you dionyse it out, or is it just left in?
I actually am not sure. I think we left it in.
Okay, moving on. You can find that out. So T-gas, a lovely question, which is in your hybrid imaging scheme. Have you experimented with minimizing the number of the tilt images so that you can put more dose into your 0-degree exposure? To what's your minimally useful tomogram?
Yeah, that's a great question. Obviously, that depends on the thickness of the sample and the size of your target molecule. So, and we were trying to be on the careful side when we approached this. So our first paper and the first structure was with 15 electrons for the untilted image, and I think about 100, 120 for the entire as an entire exposure. But however, lately we started putting more dose into the untilted image. And I think at least for the ryanodine receptor, it works rather well because it's large. And that's true that the quality of the tomogram doesn't have to be very superior, because essentially we only needed to get maybe a 15 angstrom structure, and then we go to the projections. So I think there is some wiggle room to play with.
So then there's a really interesting question, a biological question, which is from Paul, which is C4 symmetry of the receptor valid in the native membrane? And can you, for example, lose the C4 symmetry and compare to a reconstruction with a quarter of the particles?
Yeah, great question. Great question. We tried to do symmetry expansion and play with it, but we then we didn't invest as much time in it as we should. I think that potentially there would be slightly different confirmations, obviously. We need to explore it further.
Okay. And then we've got time for, there are a couple of questions which are kind of linked in my head here, which is Susan written with a similar alignment refinement strategy to Dynamo? And could you give us a little bit more detail about what's going on there? And then linked to that possibly is, how does Susan fare if the sample gets more crowded?
Well, from the algorithmical point of view, Susan is just a change of the order of operations. So from the signal point of view, it's equivalent. It's written, like the high performance part of the code is written in C. And it basically, it's inspired by the same angular convention of Dynamo. There is a very simple conversion of data, so Dynamo table can be converted into Susan data structure and back. So yeah, essentially we try to mimic the overall workflow of the project, just the whole cross correlation computation happens differently.
Okay. I think in the interest of time, we'll leave it there, and I'll thank you again for a fantastic talk. There are lots of questions for you to.
Thank you. I'll be in the chat.
Yeah.